Hi class, I'm Dr. Five. I want to talk today about genes and genetic diseases. Genes are the basic unit of inheritance, and they're composed of sequences of DNA. These are the blueprints of proteins in the body, and they influence all aspects of the body's structure and function. There's 20,000 to 25,000 genes, and an error in one gene can lead to a genetic disease. Genotype is the composition of genes at a given locus, and phenotype is the outward appearance of an individual and is the result of genotype and environment. DNA, RNA, and proteins. Genes are composed of DNA. DNA is composed of 4 types of nitrogenous bases. There's adenine, cytosine, guanine, and thymine. Commonly referred to as the first letters of A, C, G, and T. Nucleotide is a DNA subunit consisting of 1 deoxyribose molecule, 1 phosphate group, and 1 base. To serve as a basis for genetic inheritance, DNA must be able to code for all of the body's proteins. Proteins are composed of 1 or more polypeptides, which are composed of amino acids. And the body contains 20 possible amino acids. Replication. DNA must be able to replicate accurately during cell division. Complementary base pairing is pairing of adenine with thymine and guanine with cytosine, which is key to accurate replication. So those have to match up with one another. When replication is complete, a new double-strand molecule identical to the original is formed. The single strand is a template of which the complementary molecule is built, and it's the basis for synthesizing the new double strand. Mutation, inherited alteration of genetic material is considered a mutation. Microscopically observable alterations of chromosome number or structure are different examples of mutations. Some mutations are too small to be observed under a microscope. Base pair substitution. This is where one base pair is replaced by another, and this is sometimes called a missense mutation. A frame shift mutation can occur, and this is insertion or deletion of 1 or more base pairs to the DNA molecule. Mutagens, these are agents known to increase frequency of mutations, and these include radiation and different chemicals. From genes to proteins. DNA is formed and replicated in the nucleus. Protein synthesis occurs in the cytoplasm. Transport of DNA code from nucleus to cytoplasm and subsequent protein formation involves 2 processes, both of which are mediated by RNA. Transcription. RNA is synthesized from the DNA template and results in formation of the messenger RNA, and that's abbreviated mRNA. This begins when the enzyme RNA polymerase binds to promoter site on the DNA. Proteins called transcription factors bind to DNA, and these factors can activate or repress expression of genes. For translation, RNA directs the synthesis of a polypeptide. Messenger RNA cannot code directly for amino acids, so it interacts with transfer RNA, and this has a site for attachment of an amino acid. Now, RNA differs from DNA in that uracil is one of the 4 bases instead of thymine. RNA is usually single-stranded. I had on this slide back here the differences between DNA and RNA, so you can see the differences here, and that RNA has this one in the chain, and this one has thymine instead. Now, moving on to chromosomes. Human cells are categorized as gametes, which are sperm and egg cells, and somatic cells, which are all other cells other than the gametes. Somatic cells, there's 46 chromosomes in the nucleus of diploid cells, and they occur in pairs. gametes or haploid cells, and they have only one member of each chromosome pair of the 23 chromosomes. Meiosis or haploid cells are formed from diploid cells. 22 of 23 chromosome pairs, two members of each pair are virtually identical in microscopic appearance and DNA sequence, and this is considered to be homologous. 22 chromosome pairs are homologous in males and females and considered autosomes. The remaining pair of chromosomes, sex chromosomes, two homologous X chromosomes in females and non-homologous pair X and Y in males. Chromosomal aberrations and associated diseases. Chromosomal abnormalities, they're a leading cause of mental retardation and of miscarriage. Major chromosome aberration occurs in more than half of all conceptions. Most fetuses don't survive to term. About 50% of first trimester miscarriages have major chromosomal aberrations, and it's just not conducive with life. About 1 in 150 live births has a major diagnosable chromosomal abnormality. Polyploidal cells with multiple of normal number of chromosomes are euploid cells. Normal gametes are haploid, and most normal somatic cells are diploid, and both form euploids. When euploid cell has more than one diploid number of chromosome, it's called a polyploid cell. Now, some liver, bronchial, and epithelial tissues are polyploidal. Triploidy, a zygote with 3 copies of chromosomes instead of 2. And there might be tetraploidy, which is euploid cells have 92 chromosomes. Nearly all miscarried or stillborn, when they have these issues, those that survive to term usually die shortly after. Chromosomal aberrations and associated diseases and euploid cells. These do not contain the 23 chromosomes. Trisomy has 3 copies of one chromosome, and monosomy has 1 copy of a chromosome in a diploid cell. This results from non-disjunction. Chromosomes or chromatids fail to separate during meiosis or mitosis. Autosomal aneuploidy, we have trisomy, can occur in any chromosome, but commonly it's seen in the 13th, 18th, and 21st chromosomes. Trisomies in other chromosomes do not usually survive to term. Autosomal aneuploidy, we'll talk about Down syndrome. This is trisomy of the 21st chromosome, and it occurs in about 1 in 800 live births. 97% of cases are due to non-disjunction, and 3% result from translocations. IQ is generally between 25 and 70. There's distinct facial appearance in patients with Down syndrome. They have a low nasal bridge, epicanthal folds, protruding tongue, flat and low set ears. There's poor muscle tone or hypotonia and a short stature. Now, with this type of abnormality, congenital heart defects occur. There's a reduced ability to fight respiratory infections, and there's an increased susceptibility to leukemia in patients with Down syndrome. By age 40, symptoms identical to Alzheimer's disease may occur, and one of the genes that causes Alzheimer is on chromosome 21, so that's why there's a higher risk factor for these Alzheimer in patients with Down syndrome. Sex chromosome aneuploidy of 1 in 400 males and 1 in 650 females, this happens. Trisomy X, there's 3 X chromosomes instead of 2. Most have no overt physical abnormalities, but patients may be sterile, have menstrual irregularities, or cognitive defects. We have Turner syndrome, which is 1 X and no homologous X or Y chromosome, which results in 45 chromosomes. So no Y chromosomes, so only females are affected, and usually have gonadal streaks instead of ovaries. And here's a some symptoms, clinical manifestations that you're going to see with Turner syndrome. Now, Klinefelter, this is karyotype 47 and is XXY. So there's 2 X and 1 Y chromosome in each cell. The male appearance, usually they're sterile, and about half develop gynecomastia, small testes, sparse body hair, a high-pitched voice, and mental impairment. And here's information here showing a Klinefelter syndrome. Elements of formal genetics, dominant allele whose effects are observable, and we have recessive, in which the allele whose effects are hidden. A carrier is an individual who has a disease-causing allele, but is phenotypically normal. And we have autosomal dominant disorders, in which the affected offspring are produced by the union of a normal parent with an unaffected heterozygous parent. Autosomal recessive, most recessive alleles are maintained in normal carriers, and they are able to survive from one generation to the next. That's how those are passed down. Now we have X-linked inheritance. Most sex-linked traits are located on the X chromosome. And here you see it has the differences with the X-linked dominant inheritance. We're going to move on to genes, environment, lifestyle, and common diseases. Factors influencing incidence and disease in populations. Some of the terminology here, we have incidence, which is a number of new cases of a disease reported during a specific period, which is typically 1 year. And this is divided by the number of individuals in the population. Now, prevalence is the proportion of the population affected by a disease at a specific point in time. And relative risk, this is the measure of the effect of a specific risk factor. And then you can see here the formulas for that. Genetic of common diseases, we have congenital malformations. Congenital diseases are present at birth. Most have a multifactorial etiology. Examples include a cleft lip and palate and pyloric stenosis, and these are easily repaired malformations. Now, neural tube defects, these have more severe consequences. Some cases occur in the absence of other problems, but it is common for them to be associated with other disorders. There's environmental factors that can play a role in congenital malformations, one of which was a medication called thalidomide, which leads to euphocomelia, which are shortened limbs, which you can see on these children here. Coronary heart disease and hypertension, those with a positive family history are 2 to 7 times more likely to have heart disease than individuals with no family history. So family history is a pretty strong, non-modifiable risk factor for coronary heart disease and hypertension. Familial hypercholesterolemia, there's a defect in LDL receptor deficits. And with hypertension, the genes involved in the renin-angiotensin system can contribute there to hypertension. Now, when we come to cancer, there's cancers that occur strongly in families. Some of those that occur more strongly in families are breast, colon, prostate, and ovarian cancers. There's genetic and environmental factors involved here. Breast cancer, a woman with a first-degree relative of breast cancer, her risk doubles for breast cancer. And that involves the BRCA1 and BRCA2 for breast cancer. With colon cancer, a patient is 2 to 3 times higher risk with one affected first-degree relative with a history of colon cancer. Other cancers that occur commonly in families include retinoblastoma. And here's just some factors that can cause some genetic changes that lead to cancer. Genetics of common diseases, diabetes mellitus. We have type 1 and type 2. Type 1, siblings of individuals with type 1 diabetes have a substantial elevation in risk. HLA class 2 alleles, and approximately 95% of whites with type 1 have HLA DR3 and or DR4 alleles. Risk of offspring of diabetic mothers is 1 to 3%, and the risk of offspring of diabetic fathers is 4 to 6%. Now with type 2 diabetes mellitus, neither HLA associations nor autoantibodies are commonly seen. Monozygotic twin concordance rates are substantially higher than those with type 1, often exceeding 90%. And the most significant gene identified is the TCF7L2. With Alzheimer's disease, the risk doubles with a first-degree relative with Alzheimer's. About half of early onset cases are attributed to mutations in any of 3 genes: the presenilin 1, PS1, presenilin 2, or PS2, and the amyloid beta precursor protein, APP. The late onset form of Alzheimer's disease, there's allelic variation in the apolipoprotein E, or abbreviated APOE. Schizophrenia and bipolar. Schizophrenia, the risk is 10 times higher with one affected parent. The brain expressed genes dysbindin, neuroglian, diamino acid oxidase activator. And it's disrupted in schizophrenia gene DISC1. Now with bipolar disorder, there's neurotransmitter systems, and genes that are involved are those that encode for MAOA, serotonin transporter, and catechol O methyltransferase gene, which also is associated with schizophrenia. So this gene in particular here, the CMOT gene, that's associated with schizophrenia and bipolar disorder. Okay, just a little bit on epigenetics and disease. Epigenetics is a study of heritable changes in gene expression or phenotype caused by mechanism other than changes in the DNA sequence. Epigenetic modifications can cause individuals with the same DNA sequence, such as identical twins, to have different disease profiles. The case of this could be something like asthma, where you have one identical twin who might get asthma due to other factors, while the other twin does not have it because they don't have the same risk of factors associated. 3 major types of epigenetic modifications: DNA methylation. This causes genes to become transcriptally inactive or silent. The key component of X inactivation, the transcriptional silencing of genes on the X chromosome. Histone modification, chromatin structure must be controlled in self-renewing and differentiated cells in cell renewal systems. Microribonucleic acids, gene expression networks can be regulated by changes in mRNAs and other non-coding RNAs. And this is just showing different factors that can affect epigenetic modulation. And lastly, we'll talk about genomic imprinting. For some genes, the gene is transcriptionally active on only one copy of a chromosome. The other gene is transcriptionally inactive. This process of gene silencing, in which genes are predictably silenced depending on which parent transmits them, this is known as imprinting, and as transcriptionally silenced genes are then said to be imprinted. 2 examples of this, we have Prader-Willi or the Angelman syndrome. Prader-Willi, we have deletion of about 4 million base pairs of the long arm of chromosome 15. This deletion is inherited from the father. And the patient will have short stature, hypotonia, small hands and feet, obesity, mental retardation, and hypogonadism. Now with Angelman syndrome, the 4MB deletion occurs, and this disorder is inherited from the mother. Patients have severe mental retardation, seizures, and an ataxic gait. And here's just some pictures depicting those different disorders there. If you have any questions, please email me to let me know. Please keep in mind that I've only covered a small portion of these chapters from the textbook. Students are responsible for reading and reviewing and preparing all of the information for the course. Thank you all and have a great day.